Hi there, welcome to the intestinal glucose transport simulation. In this simulation, you will meet baby Jane, perform experiments to understand how glucose is transported in the intestine, and use your findings to help devise a treatment to make her better. This is the physiology lab. Here, we use mouse models to study glucose transport in the intestine. We work in collaboration with Dr. Shaw, who is a pediatrician, and today she is seeing a little patient who needs our help. So, I have arranged for you to attend the consultation, so that you can meet baby Jane and learn more about her condition.
Hi there, welcome to your first day of this research project in medical biology. I am Dr. Shaw, your clinical supervisor, and this is Jane. She has excessive diarrhea and is not gaining weight. She also has an elevated blood sodium concentration and glucose in her feces and urine. I suspect that her diarrhea is caused by her lack of ability to absorb glucose. I hope that you find some answers in the lab while you work with my colleague Dr. One. By understanding more about how the gut works at a molecular level, we can potentially apply your findings from mouse studies to humans, helping us figure out what is wrong with Jane.
Click on baby Jane to go to the lab. Welcome back. Now that you are all up to speed, I am looking forward to showing you the ropes and having a lab partner. Pick up a lab coat from the rack. Well done. Now go over to the workbench to find a glove box and click on it. I have just prepared some pieces of the jejunum section of the small intestine from a mouse, and you can find one in a petri dish on the intestine preparation workbench. I've added some ice-cold saline that needs to be topped up. Pick the bottle with the type of saline that is compatible with mammalian physiology. If you are not sure which bottle to choose, or want to learn more about the small intestine, check out the theory section. Pick up the pipette. Choose the correct solution. Transfer the liquid into the petri dish that contains the mouse intestine. Put the pipette back on its holder. Let's make an everted sac from the small intestine. Basically, this means we turn it inside out, so that the inside can be cleaned, otherwise, its contents would interfere with our results. The original interior is called the mucosal or apical side and the outer part is called the serosal or basal side. Once we obtain this everted sac, we can use it to study glucose transport across the intestinal epithelium. To get started, let's have a look at the main steps on the wall screen. I prepare everted sacs in this lab all the time. First, we need to tie one of the ends of the intestine to a glass rod or a plastic catheter. Then, we gently push the intestine to evert it, secure it with a knot, and give it a good wash. Finally, we fill it up with the desired solution, tie it shut and cut it off to release it from the catheter. Now that you know a bit more about the procedure, you get to do it yourself. Let's get started. Go back to the intestine preparation workbench and click on the petri dish containing the intestine. I have moistened a catheter with cold saline and now you will tie a piece of the intestine to the end of the catheter. Click on the intestine to get started. Next you need to gently push the intestine over the knot and over the catheter, everting it. You will also need to rinse it with saline to remove the contents of the intestine from the exposed surface. Great job, now the intestine is inside out and all clean. Let's make some more knots to close the tube. We'll start by pulling the tissue to the end of the catheter, without separating them completely, and then tie a knot at the far end. We will now fill the intestine with 1 milliliter of saline plus 10 millimole per liter glucose. But first, we need to secure it to the catheter, to make sure it doesn't come off when we start adding liquid. Click on the intestine to fill it up with saline. Well done. Now we just need to close the intestine and remove it from the catheter. Excellent. The everted sac looks slightly expanded, like a little sausage. You did a great job. If the intestine had not inflated, that would have been either because your knots were loose or the intestine was damaged, and you would need to redo the procedure. That's it. You have an everted sac. The serosal side is now facing the interior while the mucosal side faces the exterior. We are one step closer to figuring out what is wrong with Jane. The everted sac you just prepared is now in a 100 ml incubation beaker containing 50 ml of saline plus 10 millimole per liter glucose, the same as the solution inside it. To study if there is glucose transport between the two sides and in what direction, the preparation needs to be bubbled with oxygen, in the shaking water bath set at 37 degrees Celsius for 90 minutes. Pick up the beaker containing the everted sac. Place the beaker in the water bath. In a real lab, you would have to wait forever, but in virtual simulations we can speed up time. All done. Place the beaker back on the bench. Take two 15 ml tubes and place them in the rack. Transfer liquid from the beaker into a 15 ml tube. Now you have the mucosal solution. Place the everted intestine in a clean petri dish using a pair of forceps. Cut the intestine with a scalpel to release the serosal solution. Transfer the serosal solution from the petri dish into a 15 ml tube. Well done. If glucose has been actively transported by the intestine from mucosal to serosal side, then which of the following statements is correct? That's right. If both solutions had the same concentration of glucose to start with, in the absence of active transport the concentrations would have remained the same on both sides. If there is active transport towards one of the sides, then that side will have a higher concentration of glucose. To determine whether there has been glucose transport and in what direction, we need to measure the glucose on both sides of the intestinal membrane. So, we will measure the glucose concentration in the two solutions you just prepared. But first, we also need to prepare some glucose solutions of known concentration to make a calibration curve. Take the tube rack to the glucose assay workbench. I have already made 100 ml of a 50 millimole per liter solution of glucose, which you can find on the workbench. This will be our most concentrated glucose solution. Let's have a look at the wall screen to see what other solutions have prepared. Click on the wall screen. I used the 50 millimole per liter solution of glucose to make the dilutions you see on the table. To make the dilutions, I calculated what volume of the 50 millimole per liter glucose solution and what volume of water I would need to mix. I have already added the necessary dilutions into pipetting reservoirs, that we can use as known standards to create a calibration curve using the results from the spectrophotometer: 50, 25, 10, 5, and 1 millimole per liter. The final standard solution will be pure distilled water. Why do we need to create a calibration curve for the spectrophotometer before we test our samples from the intestine? Exactly. Absorbance is proportional to the concentration of a solution when on the linear part of the curve. So, if we make a calibration curve using the absorbance of solutions of known concentration, we can determine unknown concentrations of samples when we measure their absorbance if prepared under the same conditions as the standards. Do you know how the glucose assay works? Have a look at the wall screen. The glucose assay reagent contains glucose oxidase, peroxidase and O-dianisidine. The oxidase reacts with glucose to produce gluconic acid and hydrogen peroxide. Peroxidase reacts with the hydrogen peroxide produced when glucose is oxidized, thus making a brown-colored product. Since this product is unstable, sulfuric acid is added to stop the reaction by denaturing all enzymes, and a pink-colored product is formed that is stable and absorbs light at 540 nanometers. One molecule of this colored product is produced for each molecule of glucose in the solution. Wow, that was a lot of molecules and reactions. Go to the glucose assay workbench to start the assay. Safety first. You will need to wear goggles for this assay. Pick up the 5 ml pipette. Add the serosal solution to one of the empty reservoirs. Add the mucosal solution to the remaining empty reservoir. Great! Now you have all the solutions you need to start the experiment. Check out the image below to see the layout of the plate with all the solutions marked. Add distilled water to the microplate with the multi-channel pipette. Awesome! You pipetted the water into the first column of 8 wells. In a real lab you would have to pipette all the other solutions in as well, but today I will give you a hand and do that for you. Add assay reagent to the microplate. Super! As before, I will add the assay reagent to the other samples for you. In a real lab, we would need to incubate the reactions for 60 minutes at a temperature of 25 degrees Celsius. In virtual simulations, we can speed up time and get to the next step immediately. Now, we need to stop the reaction. Add sulfuric acid to the plate. You are doing so well. I love having a lab buddy. Let me pipette the rest of the sulfuric acid onto the other samples. Awesome! Now place the microwell plate in the spectrophotometer. The wavelength that will pass through the samples is 540 nanometers. You can take another look at your experimental layout in the diagram below, as well as when you pick up the plate. Pick up the microplate. Put the microplate into the microplate reader and set the wavelength to 540 nanometers. I just got the results for the absorbance. Check out the table below. Using our 6 standard glucose solutions, I have also made a plot of absorbance versus concentration, with a linear fit equation and a plot of glucose concentration versus absorbance, which you can see on the wall screen after you are done looking at the results table. The results table will be available for consultation on the Media tab of your lab pad. Click on the wall screen. Which equation should we use to calculate glucose concentration directly? Well done! The linear regression of graph B provides you with an equation that you can directly use to calculate glucose concentration by simply plugging in the absorbance value. With graph A, you would have to do an additional step by rearranging the equation to solve for the concentration of glucose. So, let's use this equation to calculate the glucose concentration in the mucosal and serosal solutions that we prepared earlier. Choose the correct values from the drop-down menus and calculate. Great! There was a concentration of 10 millimole per liter glucose on the mucosal side and 45.6 millimole per liter on the serosal side. So, was there active transport of glucose from mucosal to serosal side of the jejunum? Spot on! Both sides started with 10 millimole per liter of glucose, but now there is more on the serosal side, so active transport of glucose must have occurred from the mucosal to the serosal side. Blockers are often used to study how transporters work. So, maybe we can use a glucose transport blocker to help us understand this process and figure out what is wrong with Jane. If I remember correctly, my colleague Dr. Shaw said she presented with diarrhea, elevated blood sodium concentration, and glucose in her feces and urine. We just observed that glucose can be actively transported from the mucosal to the serosal side of the intestine. Also, the sodium-potassium ATPase is an important ion transporter in many cells, that can be inhibited by the drug Wabayan. Which of these findings using the Everted-Sack preparation would support the idea that active glucose transport is driven by a sodium-potassium ATPase on the serosal side? Precisely. If the ATPase is on the serosal side, and it drives active glucose transport, then a blocker on that side would reduce the amount of glucose there. Let's put it to the test then. I performed an experiment yesterday, where I prepared some everted sacs and added Wabe into either the mucosal or serosal solutions. Let's have a look at the results on the wall screen. Take some time to look at the results. I have also prepared glucose solutions of known concentration for a calibration curve. Since all samples were prepared in the same way, the relative values of absorbance are proportional to the glucose concentration in each sample. So, you can make an estimation of the glucose concentration in the mucosal and serosal solutions by comparing their absorbance values to those of the standards. What do these results suggest? You've so got this. From the results table for the mucosal and serosal solutions, we can see that the only case when the blocker Wabe does not reduce glucose amount is when it is on the mucosal side and glucose is transported to the serosal side. Therefore, the data confirm that a serosal ATPase has a role in active glucose transport by the intestinal epithelium. The sodium-potassium ATPase helps create a low sodium concentration and high potassium concentration inside the cell. Diffusion of potassium out of the cell through channels then creates the negative inside membrane potential. Studies of the action potential show that if the sodium-potassium ATPase is present, there is a strong electrochemical gradient for sodium entry to the cell. Conceivably, if sodium and glucose bind to the same membrane transporter protein, this electrochemical gradient for sodium could drive active sodium transport into the cell. If a sodium-glucose transporter is driving active absorption of glucose from the gut lumen into the body and the intestine, we can hypothesize that such a sodium-glucose transporter should be on the. Let's see. We can use sodium-free saline, in which sodium is replaced by methyl D-glucosamine, on either the mucosal or serosal side of the sac, to test if a mucosal sodium-glucose transporter helps drive active transport of glucose. Let's test it out. Go to the glucose assay workbench. Add distilled water to the microplate with the multi-channel pipette. Awesome, we now have water in the first column of 8 wells. In a real lab you would have to pipette all the other solutions in as well, but today I will give you a hand and do that for you. Great, now you have all the solutions you need to start the experiment, the glucose standards and the mucosal and serosal solutions that resulted from the intestinal epithelium being exposed to saline, in which sodium was replaced by methyl D-glucosamine on either side. Add assay reagent to the microplate. Super. As before, I will add the assay reagent to the other samples for you. In a real lab, we would need to incubate the reactions for 60 minutes at 25 degrees Celsius again. In this virtual simulation, we can be much more productive and speed up time to get more things done. Now, we need to stop the reaction with sulfuric acid. Add sulfuric acid to the plate. You are doing great, high five. Let me pipette the rest of the sulfuric acid onto the other samples. Awesome, now place the microwell plate in the spectrophotometer, the wavelength that will pass through the samples is already set to 540 nanometers. Pick up the microplate. Put the microplate into the microplate reader. Check out the results on the wall screen and the image below. As before, I have also prepared standard solutions of glucose, so that the glucose concentrations in the serosal and mucosal solutions can be determined. These data show that. Bingo! The fact that removing sodium from the mucosal side eliminates the increase in glucose on the serosal side is consistent with a mucosal sodium-glucose cotransporter being involved. Was your hypothesis that if a sodium-glucose transporter is driving active absorption of glucose from the gut lumen into the body in the intestine, then it should be located on the mucosal side, correct? That's right, we just saw that this transporter is indeed located on the mucosal side, which is in contact with nutrients on the luminal side of the cell. So, glucose is taken up from the mucosal side, passes through the cell and then goes into the blood via the serosal side. Based on the data we just observed, and data from other labs, have a look at the wall screen to see a model for how the small intestine actively transports glucose. Click on the wall screen. On the wall screen, on either side, you will find blue and white buttons that point to either the right or to the left. Click on the button pointing to the right to move forward and on the button pointing to the left to move to the previous diagram and explanation. Sodium and glucose bind to a protein called the SGLT1 sodium glucose transporter 1, and the electrochemical gradient for sodium drives both sodium and glucose into the cytoplasm. As you can see here, glucose and sodium enter the cell together. Glucose moves across the serosal membrane via a protein called glucose transporter 2, according to its concentration gradient. Sodium is transported by a sodium-potassium ATPase. According to this model, the serosal sodium-potassium ATPase uses the energy from ATP to lower intracellular sodium and raise intracellular potassium. Both sodium and glucose move from the mucosal side to the serosal. Potassium diffuses through a serosal channel, making the cell membrane potential negative on the inside. Let's recap and add some more molecules. Sodium and glucose are co-transported into the cell from the mucosal side and potassium exits the cell through the serosal side. Then, on the serosal side, for each sodium ion that goes out the serosal membrane, a potassium ion is exchanged back into the cell by the sodium-potassium ATPase. The movement of positive charges pulls the negatively charged chloride through the membrane via a paracellular route, between cells. Water follows the sodium chloride and glucose transport via aquaporin channels in the membranes and paracellularly by osmosis. Water follows salt. Wabain is a blocker for the sodium-potassium ATPase that prevents the fluxes of sodium, potassium and glucose that we just observed, which can explain the elevated blood sodium concentrations, and glucose in Jane's feces and urine. My colleague Dr. Shah told me that Jane has diarrhea and some glucose in her feces and urine. The SGLT1 protein that we've just learned about is present in both the intestine and kidney, working to absorb glucose. So, what if Jane has a genetic defect which means her SGLT1 protein doesn't work properly? If so, removal of what from her diet should reduce or stop her diarrhea? Indeed, this is how we can test it, since glucose is transported by SGLT1. If glucose is not being properly absorbed due to defective SGLT1, then its removal from Jane's diet should make her better. Great, I think we have a plan. I will just send our findings and suggestions to Dr. Shah through my system. Oh, I just had another idea, that we can also send to Dr. Shah. Assuming that Jane's problem is a defective SGLT1 protein, what is the most likely reason that her blood was sodium high when she had diarrhea? That's right, the intestinal epithelium is crucial in the absorption and transport of many molecules and ions, such as glucose, sodium, potassium, chloride, and water. An imbalance in glucose absorption may have caused imbalances elsewhere, where not enough water was absorbed and the result was an increase in blood sodium concentration due to dehydration. When people realized that glucose is absorbed by co-transport with sodium, it led to the introduction of a treatment that has saved many lives, oral rehydration therapy, or ORT. One of the major killers of young infants like Jane is diarrhea, often induced by infections such as cholera, and this simple treatment has really made a difference. Why does ORT therapy work? That's it. Diarrhea results in dehydration, that when extreme can lead to death. Oral rehydration therapy usually includes sodium, glucose, potassium chloride, and trisodium citrate, which stimulate their absorption, together with water. Great job! I think my colleague will be very pleased with our findings and suggestions. Hopefully, Jane will respond to the treatment and get all better. Now we just have to wait for a week and see if we were able to help. Oh, a phone call from Dr. Shah. The lab phone is sitting on the sink workbench behind you, near the entrance to the lab. Pick up the phone near the entrance.
Hi there, Dr. 1. Many thanks to you and my new brilliant project student. Results from your mouse studies did apply to humans, Jane in this case. Elimination of carbohydrates from her diet stopped the diarrhea, and now she is gaining weight and is healthy. Or it has also really helped with dehydration, and blood sodium levels are now getting back to normal.
Thank you, Dr. Shaw. It is always great talking to you, and I am so happy that we were able to help Jane. I guess you need to go back to your patients, but thanks for the update. We'll catch up next week at our joint lab meeting. With these experiments, we were able to figure out what was wrong with Jane and come up with a treatment. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.